Hello, everybody. I'm Dr. Prabhu Meghnathan from Hybridomics Life Science and Diagnostics. Thank you for CUBE for inviting me to present this talk on identification of lymphocytic changes in viral infections from CBC scattergram. The scattergram has been used widely for identifying various causes from leukemias and cancers. Let us look into the infectious pathology, especially understanding how a virus could also be predicted using the same scattergram. As we already know, the basic principle of a scattergram, and the principle is laser and the fluorescence. Using laser technology, we use forward scatter and side scatter. And fluorescence gives us information of DNA and RNA content. This, in turn, the beauty of the Mindray is that it executes SF cube technology, which in turn plots the scattergram in a 3D pattern. So which helps us to understand the cellular complexity, the DNA and RNA information, as well as the cell size. Using these three parameters, we could able to categorize the various patterns of diseases and the cell morphology. The normal pattern of these scattergrams are like the fluorescent dye, the fluorescent color plot, which you could see here is usually at the lower end. And then we have a monocyte, the violet color representation of it, and neutrophils, the blue color representation. The HFC cell, which is usually present on the upper part and which are usually scanned in a normal scattergrams. This is a normal prediction of how a scattergram looks at. We will be concentrating on the pattern and the distribution of the scattergram. Here, basically, we'll be looking into the RNA viruses of this scattergram. We are looking into three different RNA viruses, which is commonly known to us. One is H1N1. In H1N1 is also an RNA virus, which in turn has already caused a pandemic, and it also produces the similar symptoms of SARS-CoV of respiratory distress syndrome. The classical picture of H1N1 is more of a lymphopenia, and also it produces sometimes pancytopenia too. Looking at the scattergram of H1N1, you could see the pattern of distribution of the scattergram of a lymphocyte, a monocyte, and neutrophil is almost similar to that of a normal, which is on the right-hand side of your slide. The basic difference would be some amount of HFC cells, which is a little higher in terms of an H1N1. This would be a possibility of thing. These cells, when looked and under peripheral smear, look more of atypical lymphocytes with vacuolations and undulations of the cytoplasmic membrane. Dengue is also an RNA virus, the classical pattern of it. But strangely, the dengue pattern of scattergram is very well known to us and also totally different from the other RNA viruses. This is the scattergram of a dengue, which in turn where you could see a high fluorescence of the HFC cells, which is usually present on the upper part of it. And when you compare to the normal in the upper corner of your field, you could see the more these HFC cells are skewed up on a higher trend. So this is a classical pattern of a dengue. So let us also look into dengue for serial data or sequential data of representation of scattergram. As you can see, maybe the day 1 to the day 6, the HFC cell is probably increasing and decreasing in different cases, both having a similar pattern of distribution. Strangely, I would also like you to make a keen observation on the distance between the lymphocyte, the monocyte, and the neutrophil patterns in dengue and the normal scatter plots. Basic differences when you look at it when in a H1N1 is the distance between these two are almost similar to us, whereas in a dengue there is closeness of the monocytes and lymphocytes and neutrophils. Basically, the pattern why this happens is the cell size is almost getting similarity among these cases when infected. Now coming to our current scenario, where in turn a SARS-CoV-2 virus, which has created a huge pandemic to the world. This virus is also an RNA virus. Strangely, it also produces symptoms similar to that of H1N1 in terms of respiratory distress syndrome. And also, it also produces symptoms similar to that of dengue, where in turn sometimes patient presents with a platelet low count and hemorrhagic features. So looking at these pattern of the SARS-CoV with different varied patterns from coagulopathy to that of respiratory syndromes, looking at the pattern of scattergram is little much more stranger to us. When you look at it, it has the exact copy of the same of a normal distribution pattern of a normal population to that of a scattergram of a SARS-CoV-2, except for the few targeted high fluorescent like cells, which is plotted on the upper graph here. Even here, a scanty amount of high fluorescent cells. This is a sequential pattern of SARS-CoV-2, wherein a patient initial feature presents a little bit of scattered high fluorescent cells with a distribution pattern similar to that of a normal scattergrams. Whereas after a few days, you could see that there is a lacunae gap, but the site scatter part of it remains the same distribution pattern when compared to that of the first. And the high fluorescent cells population also remains almost the same. This is a characteristic pattern which we could notice in SARS-CoV-2 after a sequential pattern. When we looked at morphology of these cells looked more like a plasmacytoid-like cells or not vacuolated. The cytoplasmic membrane was more smoother, not like undulated or like an atypical lymphocyte, which is characteristically seen in other viral infections. This is another case which also produces the same pattern of initially a scanty amount of it, of the high fluorescent cells, which is almost the relatively same numbers, except for the width distribution distances from these scattergrams, which is varied or widening of the gaps is noted. Basically, all the SARS-CoV-2 do also produce lymphopenia in their action of during the course of this. Let us look at the cases which are negative for SARS-CoV, or also negative for H1N1 and negative for dengue. Strangely, when we compare it, it is almost similar to that of an H1N1 or other thing, except for the pattern of the lymphocyte distribution, which is usually having a bulge or more skewed towards the side scatter towards the left. And the pattern of your high fluorescent cells is similar to that of an H1N1 or similar to that of H1N1, except for the bulb-like basement surface of the in other viral infections, which is not noted in the normal phenomenon. Whereas in H1N1, difference is that the H1N1 and normal cellular distribution looks the same, except for the high fluorescent cells, which is usually noted in H1N1. The other viral infections, what we have noticed is the high fluorescent cells, along with the increase in the amount of lymphocytes at the base. Let us also look into non-viral entity or bacterial infection, which we could characteristically label probably as a dewdrop like pattern. If you look at an H1N1, it's just normal phenomenon, whereas a normal is just having a small distribution. Whereas in this, you could see it is like a drop, dewdrop, which is just dropping down from the top. So these, though both are different viral etiology, another part is your neutrophil population is also high. We are looking more into the lymphocyte, the pattern of lymphocyte, pattern of it. Can we really look at it? Is the HFC cell different from this? When you look at it, the degree amount of HFC cells of the site scatter is very high in terms of bacterial infection when compared to a viral infection. Other point would be highlighted is that the dew, like a dew drop, you could see just forms a small, like a dew drop, like how a droplet falls down. This is a characteristic difference for any lymphocyte population from bacteria to that of H1N1, which could have been observed by us. So in general, the phenomenon what we have noticed is that H1N1 produces HFC cell, a scanty amount of HFC cell, but the pattern of distribution of the relative scattergrams of lymphocyte, neutrophil, and monocytes is similar to that of a normal population, whereas a dengue usually have a high fluorescent cells, which is very high. Along with that, these scattergrams usually tend to merge each other. The next part of a SARS-CoV-2 is that they also represent the similar pattern of a normal distribution, except that the HFC cell is very scant in terms of SARS-CoV-2. As we could see here, hardly few populations. And the next characteristic pattern, what we noticed is that these distribution of pattern scattergrams usually widens as the day progresses in SARS-CoV-2. And the lymphocyte population morphology also is drastically different from that of other viral infections when compared to SARS-CoV-2. Thank you. This would be the end of the conclusion.